Welcome to a special edition webinar brought to you by the AKC Canine Health Foundation. I'm Samantha Wright, Program Manager with the Foundation, and today I'm talking with CHF's Chief Scientific Officer, Dr. Shila Nordone. Shila will be discussing the new 2013 OAK grants and CHF's commitment to funding research that will help all dogs live longer, healthier lives. Shila, would you begin by telling us a little bit about how research works and how CHF went about choosing next year's OAK grants?
I think I should start with talking about how individual studies are important in the big picture for canine health. Typically, we look at individual studies as if they're working in isolation, but in reality, they are part of the bigger picture of understanding a disease. We know that disease is very complex. Different cells are involved, different tissues are involved, and ultimately, to really gain a deep understanding of disease with all its complexity, we have to look at certain aspects of disease in isolation. But over time, what happens is each study pigeonholes into the next study, and we develop a full picture of the complexity of disease that helps us ultimately move to better treatments and better diagnostics. Once a study is finished, the next important step is dissemination of study results. And so the way in which that happens is studies are written up as papers and they're submitted to journals, peer-reviewed journals, where the results of a study are reviewed by experts in the field. These are known as peer reviewers. When results are deemed to be important in that field, they are accepted for publication and ultimately are published in the peer-reviewed journal. And then once publication occurs, others in the field can look at the data, and the data are used ultimately to make progress in that field. I think a very important question that should be asked by our donors and by our supporters is the Canine Health Foundation contributing to scientific and medical progress? And I can tell you, absolutely we are. To date, we have 114 publications in peer-reviewed journals, so this is as of October 2012. And most importantly, these 114 papers have been cited 589 times. So that means that people have picked up on the study results and have used them to go to the next step. So what this means collectively is that the results of our funded research are being used to an incredible extent within the research community. The process of choosing research that merits funding, I think, also needs to be discussed. We start our process in January. So January of 2012, we put out a call for pre-proposals, and we received 160 pre-proposal applications. We reviewed these internally using the Canine Health Foundation's internal grant review committee, and they felt that 82 of these 160 applications had potential high impact in canine health. So those 82 applications were invited back as full proposals. Once those full proposals came back, they were sent out to 80 peer reviewers, so experts working in the field to study. And when those reviews came back, we found that 39 of those 82 applications were deemed to be scientifically meritorious and of high impact to the field. The total requested amount from the 39 applications that came back as being of high scientific merit had a total dollar amount of $4.4 million, and our budget this year was $1.8 million, which meant that we had to make some decisions about funding priorities. So the factors that influence our funding decisions are, first and foremost, being responsive to our donors. Second, covering the breadth of canine health concerns that are out there. Third, we need to be directive in some cases with emerging health concerns. There are some health concerns that our supporters and our donors may not be aware of yet. And then the last thing is that we need to create depth in our funding portfolio with some of the major health concerns. And I'll talk about depth versus overlap at the end of this talk. And ultimately, in the end, we funded 17 OAK grants for this year. As I'm going through the new research proposals that we funded, I'm going to be talking about them in the context of program areas. One of our goals is to have balance in the program areas we fund so that we don't overlook cardiology for ophthalmology or get particularly weighted in one area versus another. So if you go to our website, you'll be able to see all the different research program areas that we have now put in place.
Now that we understand a little bit more about the research process, would you tell us about the new grants?
I'm going to start with the cardiology research program, and our first grant in this research program area is 1753, Identification of Genetic Factors that Alter the Severity of Cardiomyopathy. This grant was awarded to Dr. Kate Muirs at North Carolina State University. And what Dr. Muirs is going to do is determine whether or not there are additional genetic factors present that affect cardiomyopathy in dogs that have a deletional mutation in the striatin gene. Ultimately, what her work will do is improve our use and interpretation of the genetic test for the striatin mutation. So she's taking things another step farther and helping us understand essentially the complexity that can come with genetic-based cardiomyopathy. The next grant in our cardiology research program area is 1760, Use of Gene Therapy to Treat Dilated Cardiomyopathy. This was awarded to Dr. Margaret Sleeper at University of Pennsylvania. This is a really exciting grant because it is getting us into the realm of using gene therapy to treat some of our genetic-based heart disease in dogs. So the focus of Dr. Sleeper's research is dilated cardiomyopathy. It's the second most common cause of heart disease in dogs. What she is going to do is conduct a placebo-controlled double-blinded study to evaluate gene therapy approaches in the treatment of dilated cardiomyopathy and congestive heart failure. Her approach that she's using in this study is focused on addressing the problems that we have with malfunctioning calcium channels in heart disease. So ultimately, her work has broad ramifications for all dogs with heart disease because malfunctioning calcium channels are common in most heart disease in dogs. I'm going to switch gears now and talk a little bit about the research we funded in our musculoskeletal research program area. The first grant is 1762, Use of Plasma-Derived Growth Factors to Heal Cruciate Rupture. This was awarded to Dr. Peter Muir at the University of Wisconsin-Madison. So in this study, Dr. Muir is taking a novel regenerative medicine approach to the treatment of cruciate rupture. He is going to be treating dogs with platelet-rich plasma. And this is derived from the dog itself, not from different dogs. And the belief is that there are growth factors in platelet-rich plasma that can help promote healing of cruciate ligaments. And so this, to some extent, is being used right now in private practice, but we don't have any evidence-based protocols for using this. There's only anecdotal evidence out there that PRP treatments do work for craniate cruciate ligament disease. So this will provide us some evidence-based proof that this is indeed the way to approach treating cranial cruciate ligament rupture. The next grant in our musculoskeletal research program area is 1782, Defining the Elements of Successful Cranial Cruciate Ligament Repair. This was awarded to Dr. Gina Bertacci of the University of Louisville. Dr. Bertacci is actually a biomedical engineer, and so she comes at cranial cruciate ligament disease from a different approach. She has developed a 3D computer model, and what she's going to do is use this computer model to improve our understanding of stifle or knee biomechanics. Following surgery, the surgeries that we typically use to repair CCL disease, she's also going to use this computer program to investigate the anatomical characteristics of the knee to gain an improved understanding of their role in the outcome of surgical intervention. So ultimately, this study is going to be a biomechanical approach to develop an evidence-based assessment of the current procedures that we use to repair knees in dogs. Now I'm going to switch gears and talk to you about a grant we funded in the context of our neurology research program area. It's 1731, a novel approach to understanding how meningoencephalomyelitis develops in dogs. So this was awarded to Dr. Nick Jeffrey at Iowa State University. Meningoencephalomyelitis of unknown etiology, or MUE, is a very long way of saying inflammation of the brain and spinal cord. We don't understand what causes MUE, but we do know that the onset is acute, happens very quickly, and can lead to paralysis, seizures, and is also rapidly fatal. We also know that small breeds such as miniature poodles, Maltese, Dachshunds, West Highland White Terriers, and others seem to contract MUE at a higher frequency than larger breed dogs. So there's some recent experimental evidence that bacteria in the digestive system are triggers for MUE, and this has been found in the rodent model and in mice and rats. And so what Dr. Jeffrey is going to do is determine whether imbalances in the number or types of digestive system bacteria in the dog are potentially a cause for MUE. And this ultimately is going to lead us to new approaches for treatment of MUE in dogs. Now I'm going to get into oncology. The Canine Health Foundation has had a very extensive history of funding oncology research, and so we did fund several oncology grants this cycle. And I'm going to start with the grant that's 1843, Investigation of the Genes Controlling Canine Leukemia to Properly Diagnose and Control Disease. This was awarded to Dr. Matthew Breen at North Carolina State University. And consensus opinion is that the true incidence of leukemia in dogs is unknown and that many cases highly likely go undiagnosed. Dr. Breen has been working with leukemia for several years, and he's found that the disease in dogs is characterized by chromosomal and genetic changes that are highly similar to those observed in humans. And so what he's going to be doing in this study is screening a large cohort of canine leukemia patients for the presence of recurrent chromosomal and genetic changes. Ultimately, this study is going to help us define the pathogenesis and improve our diagnosis and treatment of canine leukemia. The next grant is 1822, Beyond the Genome, the Intersection of Genes and the Environment in Canine Cancer. This was awarded to Dr. Robert Wayne at the University of California, Los Angeles. I think it's the most important thing for our supporters to know and to understand is that not all genes are active at all times. And we know that DNA methylation, or the addition of methyl groups to DNA, is one of several mechanisms that the body uses to control gene expression. Understanding of DNA methylation patterns have not been established in the dog. They have been established in people, and we do know that abnormal patterns of DNA methylation are observed in human cancer. So what Dr. Wayne is going to do is develop the techniques and methodologies that we need to start exploring DNA methylation, or the methylome, in the dog. Ultimately, this is going to become a new resource for understanding how gene regulation occurs through methylation, and is going to have broad effect across all different types of canine disease. The next grant in our oncology research program area is 1826, a novel treatment of brain tumors using a one medicine approach. So this is a one medicine grant because it is combining both human and veterinary medicine. We have a veterinarian, Dr. Simon Platt, working with a human physician, Dr. Kostas Hadjipanayas. Dr. Platt is at University of Georgia, and Dr. Hadjipanayas is at Emory University School of Medicine. The two together have developed a very novel drug delivery system that they've coupled with a novel therapeutic agent that directly targets brain tumors. They're going to be administering this novel treatment to dogs with gliomas, or brain tumors. They're going to be clinically monitoring these dogs over time, over 12 months, using MRI. And ultimately, they're going to be looking at progression-free survival of these patients and reduction in tumor size. So this is a very unique grant with a unique team of investigators. The next oncology research program area grant is 1759, Disrupting the Differentiation of Cancer Stem Cells to Prevent the Spread of Hemangiosarcoma. This was awarded to Dr. Jaime Modiano at University of Minnesota. Hemangiosarcoma is a devastating disease. It's rapidly fatal. And what we do know about it is that it does seem to adhere to the cancer stem cell model, in that there are cells involved in hemangiosarcoma that constantly reseed the tumor and contribute to its spread or metastasis. Dr. Modiano has a very interesting hypothesis. He thinks that if he can force these tumor stem cells to terminally differentiate into cells that can no longer self-renew, he can disrupt the ability of hemangiosarcoma to spread. And he also believes that in doing this, he'll be able to enhance the sensitivity of these cells to conventional and targeted chemotherapeutics. The next grant is 1787, Clinical Advancement of a Cancer Vaccine in Dogs. This was awarded to Dr. Nicola Mason at the University of Pennsylvania. And this is actually a competitive renewal of a previous grant funded to Dr. Mason. This grant focuses on canine lymphoma, which is the most common blood-based cancer in dogs. In a previous study, she developed a cell-based vaccine that induced anti-tumor immunity in dogs with lymphoma. And the initial studies using this cancer vaccine were very hopeful. She found in her early studies that the vaccine significantly prolonged second remission duration and overall survival. But unfortunately, the vaccine did not prevent relapse. So in this study, she'll be optimizing her cell-based vaccine approach. And her goal is to induce functional, long-lasting tumor-specific immune responses that will ultimately prevent relapse and prolong survival in dogs with lymphoma. Our last grant in our oncology research program area is 1806, a novel virus-based anti-tumor treatment for canine osteosarcoma. This was awarded to Dr. Bruce Smith at Auburn University. We know that osteosarcoma is an incredibly aggressive canine bone cancer, and the prognosis is very poor. Fewer than 20% of dogs survive up to 2 years. So what Dr. Smith has done is developed a novel virus-based anti-tumor treatment that he has preliminary data showing that this virus-based anti-tumor treatment kills tumor cells but spares healthy cells. And so through this study, he's going to be evaluating the efficacy and trying to understand the mechanism of action of this anti-tumor treatment. Now we're going to move into our renal disease research program area. And the first grant in this program area is 1766, Identification and Validation of the Genes that Define Abnormal Development of the Kidneys in Dogs. And the person that this grant was awarded to is Dr. Kirsten Lindblad-Towe of the Broad Institute. The focus of her research is renal dysplasia, or the abnormal development of the kidneys. We know that we have an increased prevalence of renal dysplasia in certain breeds, such as boxers, miniature schnauzers, Shetland sheepdogs, and some other breeds. And this suggests that there is an underlying genetic influence. So what Dr. Lindblad-Towe is going to be doing is conducting genetic and functional studies to identify the causative mutation that leads to renal dysplasia. Ultimately, what we'll be able to do is improve our understanding of renal development, and we'll be able to hopefully develop breed-specific genetic tests to prevent the disease. The next grant in our renal disease research program area is 1844, Treatment of Urinary Incontinence with Multipotent Muscle Cells, a regenerative medicine approach to a common canine health problem. This grant was awarded to Dr. Shelley Vaden at North Carolina State University. We know that urinary incontinence affects more than 20% of spayed female dogs and affects medium and large breeds to a greater extent. What Dr. Vaden is going to do is inject cultured muscle cells with the hopes of restoring function of the urethral sphincter in dogs with incontinence. And so collectively, what she's going to be doing is developing a novel regenerative medicine approach to start treating a very common canine health problem. Now I'm going to talk about a grant that falls under our general canine health research program area, and that is 1827, Defining the Specific Species of Bacteria that Contribute to Canine Periodontal Disease. This was awarded to Dr. Marcello Riggio at the University of Glasgow. So we know in humans, consensus opinion is that specific bacteria cause gum disease in humans, but we know very little about the underlying cause of gum disease in dogs. It is a prevalent problem with a high degree of pain associated with it. What Dr. Riggio is going to do is use a novel technique called high-throughput deep sequencing, where he's going to be able to define and understand all the different types of oral bacteria in dogs with gum disease and without gum disease. Ultimately, what this is going to allow us to do is begin the process of developing vaccines and improving treatment methods for dogs with periodontal disease. A grant we funded that has broad application across several different research program areas, immunology, infectious disease, and oncology, is 1771, Defining the Unique Genetic Markers in Dogs that Define Immune Function, Disease Resistance, and Tissue Transplantation. This grant was awarded to Dr. Aravind Ramkrishnan at the Fred Hutchinson Cancer Research Center. And the focus of his research are the major histocompatibility complex genes, or MHC genes. These genes encode proteins that are critical for a wide range of biological functions, from immune protection against infectious disease to ultimately the predisposition of an individual to develop diseases such as diabetes or autoimmune disease. We understand a great deal about the MHC in people, but we know very little about the MHC in dogs. And so what Dr. Ramakrishnan is going to do is use high throughput sequencing in order to define MHC genetic variation in over 1,200 dogs from at least 50 different breeds. Ultimately, what this is going to do is allow us to start doing stem cell transplants from dogs that are potentially not related, and it's also going to take us into the realm of potentially tissue transplantation in dogs. So collectively, this will have a broad impact across all of canine health, including oncology, immunology, and infectious disease. Our next grant falls under the category of infectious disease research program area, and it's 1780, defining the mechanism by which ticks locate dogs in order to better prevent disease transmission. This was awarded to Dr. Emma Weeks at the University of Florida. And this is a very interesting study. We know that the brown dog tick is the most widely distributed tick in the world, and it carries with it a host of pathogens that can lead to diseases such as ehrlichiosis and babesiosis. They can actually cause infestation indoors and are incredibly hard to control because of pesticide resistance. So Dr. Weeks' hypothesis is that if we understand what it is about the dog and the chemicals secreted by the dog that attract the brown dog tick, we'll be able to manipulate tick behavior and ultimately manage and reduce the need for extensive use of pesticides in the environment. Our last grant falls under all of our research program areas, and that's 1849, Filling the Gaps in the Canine Genome. This was awarded to Dr. Sheng Zhao at the University of Georgia. So we've had the canine genome available for 8 years now, and we're actually in our third iteration of the canine genome, CanFam 3.1. The problem is the gene annotation or differentiation and identification of coding sequences has not been thoroughly done. And so we know that we still have substantial errors in the genome and that we're missing dog-specific information that really significantly hinders research in several different fields that don't rely on the identification of SNPs, but rather need the whole coding sequence for genes. So what Dr. Zhao is going to do is use state-of-the-art next-gen sequencing strategies to more completely annotate the canine genome. And as I said, this has application across all aspects of canine health. And she's going to be doing this in a variety of major tissues and cell types. So we'll get a great deal of information out of this grant.
How will all of these grants affect CHF's research portfolio?
So I think we need to talk about depth in our funding portfolio and make the discrimination between depth and overlap. The goal of our funding program is to fund stepping stones. And those stepping stones ultimately, we hope, will fill our gaps in knowledge. So there's a difference between funding multiple grants in a program area so that you create multiple stepping stones and having overlap or grants that are essentially doing the same thing. We don't have grants that are doing the same thing. They're all coming at problems from very different angles. So I want to give you an example of program depth. And I'm going to use cranial cruciate ligament disease as the example. And I'm going to be talking about grants that were newly funded and also some grants that are active in our grant portfolio. We know that cranial cruciate ligament disease is an important problem in dogs. It costs us $1 billion a year and affects 2.5% of all dogs. So if you look at cranial cruciate ligament disease as one problem that we have, we're coming at it from different angles. The first grant would be 1762 that I just told you about, where Dr. Peter Muir is using platelet-rich plasma as a potential treatment for cranial cruciate ligament disease. The next grant is 1856. This was recently funded to Dr. Mandy Lopez, and she's trying to identify the best stem cell source as a potential treatment for disease. I mentioned to you Grant 1782, Dr. Gina Bertacci's grant, where she's using computer modeling to understand the best surgical approach and understand how the biomechanics of a dog actually influence disease and ultimately surgery outcome. And then finally is Grant 1584, in which Dr. Dominique Griffin is creating confirmation scores. Her hope is to be able to create confirmation scores that'll lead to an understanding of which dogs ultimately may be susceptible to cranial cruciate ligament disease. But what you can see is we've come at the disease from different angles so that we can create a program of research that addresses both underlying causes, the best surgical approach, and hopefully the best treatment options to treat disease. So the last thing I want to talk about is funding of research. And for our supporters, as they're looking through the grants, from here on out, what they're going to be doing is sponsoring grants in the context of a program area. And what that means is they get to receive all the progress reports within that program area. And they can also know that they are contributing broadly to the stepping stones that will help us prevent, treat, and cure canine disease.
Thank you so much for all of that great information, Sheila. To learn more about the exciting research discussed today, or to make a donation to a grant or program area, please visit us online at www.akcchf.org or give us a call at 888-682-9696. Thank you for listening and for your support of Canine Health.